Great, thank you for that. My name's Daniel Dashevsky, and today I'm going to be talking about the venoms of Australian spider wasps from the family Pompilidae. I just want to start out by thanking everyone who made this research possible. CSIRO is funding my position in the Australian National Insect Collection, where I work as part of the Hymenoptera lab in Juanita Rodriguez's group, and she'll be giving tomorrow's plenary. We also have a lot of collaborators, including Andy Walker and Sam Robinson at the University of Queensland. We have other members of the Hymenoptera lab, including Maddie Giannata and Uday Mawala-Gadero, who spoke earlier in this session. My good friend James Dobson, who helped us do some field work up in Cooktown, which was enabled by the Yuku Bajamaliku people, who gave us access to their lands and showed us around and really helped us with that collecting trip. I also want to acknowledge the Ngunnawal and Ngambri people of the Canberra region, where I live and work and where we've done additional field work. And of course, I also want to thank all the conference organizers for making this happen. So I'm talking about parasitic wasps. Most of you are probably pretty familiar with this idea. It's a very ancient lifestyle. The first parasitic wasps probably evolved somewhere in the Permian. And there have been some secondary losses, such as the social bees and ants and wasps. But for the most part, most hymenopterans are still parasitic. And as we know, this is one of the hyperdiverse orders of insects. So it's estimated that there's maybe something like 700,000 species of parasitoids. And this means that probably a sizable plurality of all the venomous species on Earth are hymenopterans. I'm going to be talking specifically about the pompilids, which are a family of spider specialists. So they will sting a spider to paralyze it. They will lay their egg on it, and the larva will then consume that spider as it develops. They're globally distributed, found from tropical to temperate regions on every continent except Antarctica. And they're really important for a lot of ecosystems. Not only are they obviously intimately connected with spiders, who are themselves keystone species, but they're also pretty significant pollinators, because while the larvae feed on the spiders, the adults themselves are actually nectivorous. And so as they go from flower to flower to nourish themselves, they'll also transport the pollen around. But I'm going to be looking specifically at their venom. And this obviously has a major function is to help with the parasitism. So it paralyzes the spider. And then it also potentially acts as a bit of a preservative, because as you can imagine, if you've got a spider stored underground for what could be up to months on end, depending on the species, with open wounds and like the larva feeding on it, there's a lot of opportunity for decay and other detritivores to set in and sort of spoil some nutrients that could go to the larva. So it makes sense that there might be some role for the venom to play there. And then they're also used as defensive tools for the adults. These stings are supposed to be incredibly painful. The large species, such as Pepsis, the tarantula hawks from the Americas, and I also hear the really big ones here in Australia are comparable. They are rated as a 4 on the Schmidt Sting Pain Index, which is the highest rating that these insect stings can get. So they're up there with things like the bullet ant, though luckily these stings are supposed to not be painful for that long. And the cool thing about venoms is that there's a lot of variation there, and it's a really interesting system to study in terms of how phenotype and genotype are connected, because it's obviously highly functional. There's a lot of selection pressure on it, and it's a very simple gene expresses as toxin. Toxin is introduced to the target. There's not a lot of interaction going on, and it's pretty straightforward. The pompilids tend to specialize by ecology more than species, so they'll have things that feed on. This is a Pepsis that's going after a tarantula, but it might take a big wolf spider that's also on the ground. You have other taxa that go after spiders and webs or jumping spiders like this little guy. So they're not necessarily like specializing by the species of their hosts. So we're not necessarily expecting that much taxon specificity in the venom, but you get other axes of variation, such as their actual parasitization strategy. Some of them will simply sting the spider and let it walk away freely rather than digging a burrow. Whereas obviously others of them do dig the burrow, but some of them will dig the burrow before stinging the spider. Some of them will dig the burrow afterwards. And as you can imagine, this all sort of changes how long that spider needs to be paralyzed for. It might be, if you want it to walk away, then the less time it's paralyzed, the better, because it has to sort of try and keep itself alive to keep your larva alive. If you're going to just stick it straight in a burrow, maybe that'll constrain it and it doesn't have to be paralyzed that long. And then some will even just chew the legs right off the spider. And then once it doesn't have any legs to free itself, it doesn't matter how long the venom paralyzes it for. There hasn't been a ton of research on these venoms, but there is some. And so we know a handful of toxins going into this. The most famous are probably the toxins, which are obviously named after the family. There's these short linear peptides that have the, you can see the basic amino acids, arginine and lysine, R and K in red. Those sort of are at a specific characteristic spacing. And those are known to mess around with the sodium channels of the muscles, possibly help to paralyze the prey or immobilize it initially, and a slower acting paralytic toxin comes in, which is what some people think the arginine kinase might be. That second bullet point there that was isolated from a big black and orange Japanese species called Phenomics fulvognathus, and there's some experimental evidence that that's actually what paralyzes the spiders from that venom. There's also anoplin, which is another small peptide. It seems to form a sort of helix that likes to punch holes in membranes, and that can be painful or antimicrobial. It's a little unclear what role it's actually playing. There's other even more enigmatic toxins, from kinins to Kunitz series protease inhibitors, and there's some enzymatic evidence for the presence of metalloproteases or hyaluronadases, which is not too surprising because those sorts of proteins are pretty common in the venoms of venomous critters from across the animal kingdom. So wouldn't be shocked if we find those too. So our first steps, we're going to do some basic phenomics, which is basically combining de novo transcriptomics and proteomics with a mass spec. So using those two data sets together to sort of reinforce each other. And that gives you a really good idea of what's expressed in the venom. It's sort of low-hanging fruit, but it's a necessary foundation for some of the more interesting studies that we want to do later. And on the screen here, we've got a venom system from a pompilid that I dissected out of one of our specimens. You've got a stinger on the left, then you've got a venom duct, which I broke during the dissection, but it's supposed to be one continuous tube that leads to that muscular little softball, which is the venom reservoir. And you can see then coming off, connected by those tiny threads, are the actual venom glands, where the little pom-pom things, where the actual venom toxins are transcribed. So I've highlighted here on this pompilid family tree in yellow which species we actually have samples from for now. So that's venoms and glands. We've got a number of samples from the Pompilus Turnomaya tanostega species group. So that should give us a good idea of what the variation looks like between closely related species. We've got samples from the sort of enigmatic basal Pepsinae over there in green. So in that subfamily, there's these something like so-and-so Kyrgyz genera, where not a lot is known of them. And we've got venom and glands from a couple of those. We've got venoms and glands from the Cryptochilus and Heterodontonyx, big orange Australian species that my lab mate Maheen will be talking about their systematics on Wednesday. And then some other species. So we've got a pretty good sampling, but obviously there's room to grow. And so hopefully this summer we'll get some more samples and sort of fill out a little bit farther on this family tree. But starting with good representation from both subfamilies, it's a strong start, though obviously the pandemic has sort of slowed everything down. And we've only got one transcriptome so far. That's from Pompilus cinereus, also called the leaden spider wasp. It's a very small species, but it's highly cosmopolitan. It's found basically from Australia to Britain, everywhere except the Americas. And it's actually coincidentally the type species of its genus, its subfamily, and the family as a whole. And you can find it pretty much anywhere there's a warm patch of sandy habitat. This particular individual that we sequenced was from some coastal dunes near Cooktown. And this species is also the target of some ongoing genomics efforts in the insect collection. So we've got some This is obviously preliminary analysis of the transcriptome, so we're hoping to refine these results and clarify exactly what they mean. But we've got some contigs that match known toxins that I've been talking about, such as the toxin, that arginine kinase that might be paralytic, and some Kunitz protease inhibitors. And the interesting thing is these are extremely conserved. So venoms in a lot of other taxa are evolving very rapidly, but these seem to be highly conserved. Think the toxin differs by one amino acid. The arginine kinase is about 355 amino acids long, and of those, it varies from the sequence from phenomics, which is in the other subfamily, in the pepsinase subfamily, rather than the pompillinase subfamily. They're only 4 amino acids different. So they're very similar toxins. We'd expect them to be pretty similar biochemically. Oddly, my first round of expression analyses showed that all these known toxins tend to be at pretty low expression levels, but hopefully we'll clarify the highly expressed toxins are not, don't really match to anything that's been characterized. So unclear if they're housekeeping or random errors, and hopefully our proteomics will help clarify this. I'm getting a time warning, so I'll try and speak quickly. Basically, the toxin from Pompilus cinereus, it turns out, has already been tested by another study that was just doing a function analysis and doing some little tweaks. It looks like it acts about the same as the alpha toxin, but is slightly less potent. So that's cool. We already sort of know what that does. And some preliminary proteomics suggest that this is one of the major toxins from the Pompilus cinereus venom. We've also got some more sequences available from a transcriptome made available by our collaborators, as well as genomes that are being sequenced in the collection. So the Botticellius vespoides and the heterodontonics sequence there. So we can hopefully maybe sequence some of those toxins and test theirs. We've been finding arginine kinase and kinins in these others. The kinins are interesting in that they're not actually painful themselves, but there's some experimental evidence to show that they make other things more painful. So that could help explain part of why the stings are as bad as they are. And then there's some weird ideas about what these Kunitz peptides might be doing, but no one's really sure. So obviously we'd like to get more samples. We'd like to synthesize some of these toxins and do some in vitro tests to characterize them, and then maybe get to some in vivo tests on spiders to see what they actually do in the natural targets. We want to integrate this with genomics data from the collection. And now when we've got really good data from our fresh specimens, we want to try and look at what we can get from preserved specimens in the collection to see if we can sort of open that up as a biodiscovery resource as well. And with that, I will take any questions. Thank you.
Okay, thanks, Daniel. That was a fascinating talk. Do we have any questions? I can start. Do you have any predictions about the evolution of some of the toxins? Do you have any thoughts about whether there might be any correlation with climate or habitat or anything like that?
I would expect some of these, like I was mentioning earlier, where the toxin duration or the paralysis duration changes a lot across these across the taxa. I would expect that that is probably something that is controlled by the toxins. It could potentially just be controlled by dosage, in that we often see the larger wasps produce longer paralysis, and that there's a dose response curve when people have done experimental tests with spiders. But larger wasps also take larger spiders. So I imagine that they're not necessarily actually getting like that much more of a dose. And that if you want a longer paralysis, it probably has to just be a longer acting venom.
Okay, cool. Interesting. Are there any, oh, there is a question. Kate, do you want to read it out, Daniel?
Yeah, so Kate asks, why are many of the toxins peptides rather than chemical in nature? Part of that is just the spotlight effect, or sort of street lamp fallacy, where we have genetics and proteomics stuff that makes it easy to analyze and synthesize and understand genes to proteins. So you can get a transcriptome and see, this transcript translates into this peptide. If I synthesize this peptide, this is what it does in the lab. But also what you find is that there are already a lot of peptides and toxin and proteins in organisms that can be co-opted pretty readily for venom function. So like there's already a huge library of functional peptides to be sort of mined from an animal's physiology. And I think there's just fewer small molecules that might do weird things to the targets.
Awesome. That's so interesting.